स्टूडेंट्स, आज हम जानेंगे कि माइक्रो ऑर्गेनिजम्स क्या होते हैं। माइक्रो ऑर्गेनिजम्स बेसिकली वो ऑर्गेनिजम्स होते हैं जो बहुत स्मॉल होते हैं और जिन्हें हम अपनी नेकेड आई से नहीं देख सकते। उन्हें देखने के लिए हमें माइक्रोस्कोप की जरूरत होती है। ऐसे स्मॉल माइक्रो ऑर्गेनिजम्स को हम माइक्रोब्स भी कहते हैं। बेसिकली 4 टाइप्स के ज्यादा हार्मफुल माइक्रो ऑर्गेनिजम्स होते हैं। फॉर एग्जाम्पल बैक्टीरिया, वायरस, फंगस और पैरासाइट। ऐसे ही और वीडियोस और शॉर्ट्स देखने के लिए लाइक करें, कमेंट करें, शेयर करें हमारा चैनल।
Today I am taking a class on contamination, preservation and spoilage of the food. Okay. So as you know that microorganisms are present in all around us, and definitely they will give the effect to our food content also. And microorganisms are so we can say that microorganisms are associated in variety of ways with all of the food that we eat. They may influence the quality, ability, quantity of our food. And the major thing is that they are naturally occurring foods such as fruits and vegetables. They are normally contain some microorganisms and may be contaminated with additional organisms during the handling of all these things. So the food can be served as a medium for the good fall and medium for the growth of microorganisms and their growth, and may cause the food to undergo the decomposition and spoilage also. Got it? So food may also carry pathogenic organisms and result that can transmit the disease also, and some other microorganisms that allow to grow in the certain food products. They can produce the toxin substances, and that is result in the food poisoning when the food is ingested. And other microorganisms that are used for the preparation of the different type of food products such as yogurts and curd. So that is also associated with the formation of food. Microorganisms are we are using for the preparation and preservation of the food also, like yogurt and other things. And generally we can say that major concern in this because of we have to discuss about the food microbiology. That means normal flora of the food, their significance, their manner in which the food can be protected, and microbial contamination, microbial spoilage, and the things by which the food can be affected. So the microbial flora of the fresh food we can discuss here. So first we have started with the introduction also. Mainly we will see the different changes in cereal and food products. Important food resources are livestock feed worldwide. The exterior and harvested grains retain some of the natural flora. Then while growing, plus contamination from the soil and insect and some other sources. So the freshly harvested grain that contains the load of thousands of microorganisms and mold also. So for the preservation, what we do, we will do the removal of the excess moisture in the cereal, grain and flours and other things, and preserve our food as a dry thing. So due to the drying of this process, the microorganism cannot grow on this, so we can avoid the spoilage and contamination of the food. Okay. So main microbial fresh food. First we will study about the main microbial flora of the fresh food. Okay. So inner tissue of the healthy plants and animals, they all are the free of microorganisms. And the surface of raw vegetable and meats are contaminated with variety of organisms. You should know about that. And the magnitude of the microbial contamination that reflects more or more of the formation of the microbial population of the environment and from which the food are taken. The condition of the raw product, the method of handling, how we are handling the food, the time which time we are collecting the fruit, and the condition on which we are storing our food products. Okay. So it is desirable to maintain the very low microbial level of contamination on the raw food and the presence of extremely large number of the microorganisms, that some undesirable events can occur and that the food is indeed susceptible to further deterioration. Okay. So main food products are the cereals, meat, poultry, eggs, fruit and vegetables, and shellfish and finfish, milk, and some other biochemical type of bacterial milk. Okay. Nowadays a lot of things are going on, and how we can characterize the bacterial retention in the milk, and different type of reactions, and now what kind of organisms that can contaminate the milk flora, and how we can preserve all these are food products, and what are the spoilage that we will discuss in further in this chapter. Okay. So first I am starting with meat. So the carcasses of healthy animals, that is of slaughtered by the meat and held in the refrigerated room, and likely to have only the normal surface contamination, and while the inner tissues are sterile. Okay. When the fresh meat cut from the chilled carcass, its surface contaminated with a microorganism and characteristics of environment, and the implement, that is saw, and the knife that is used to cut the meat. Each new surface of the meat results from the new cut and add the more microorganisms to expose the tissue. The ultimate in providing the new surface and potential contamination of the meat surface occur in this process of making the meat as a food. So, to improve the microbial quantity of the meat, particularly ground beef, and most states have to adapted standards, and that are considered established the regulation to require the microbiological standards, and the things which we had purchased from different type of outlets. Among the most common species of the bacteria that are present on the fresh meat are Pseudomonas, Staphylococcus, Micrococcus, Enterococcus, and Coliform. And at the low temperature, where the fresh meat are held, that favors the growth of phycophilic microorganisms. So that is about the meat. And second, we can discuss about the poultry. So the poultry, the freshly dressed, the poultry have a bacterial flora of their own surface, that is of the skin, and that originates from the bacteria normally present in the live birds and from the manipulation during the killing, defiguring, and aviations. And under the good condition, under good sanitary conditions, the bacterial count are reported very less bacteria per square centimeter of the skin surface, where where under the less sanitary condition the count may increase 100 times folds or more. So main organisms which are present on the egg and the other poultry things are the Pseudomonas. That is a major contaminant of the skin on the freshly dressed poultry. Okay. Next is the egg. So the interior of the freshly laid egg is usually free from microorganisms. Okay. Microorganisms effect are very in the minimal amount, and that is also depends on the sanitary conditions, which is the condition of the storage, what is the temperature, what is the humidity. Microorganisms, particularly the bacteria and the molds, that may enter into the cracked egg or in the shell or penetrate the shell when the bloom, that means the thin protein coat that is called the bloom, that is covering the shell deteriorates. Okay, the type of microorganisms that involve the reflect those present in the environment. Next is a fruit and vegetables. So fruit and vegetables are normally susceptible to the infection by bacteria, fungi, and viruses. And microbial invasion of the plant tissue can occur during the various stages of the fruit and vegetable development. And that's why the tissue are invaded and spoilage is very much increased. And the second factor that contributing the microorganism contamination of the fruit and the vegetable, that pertains to their post harvest handling. Okay, and mechanical handling is also produced, that is breaks in the tissue also. The pH of the fruit is relatively acid, that is ranging from the 2.2 for the lemon and 5 for the bananas. This restriction bacterial growth but does not retard the fungal growth. The pH that range from for the vegetable is slightly higher, that may be the 5 to 7, and that's why these are more susceptible than fruits to attack by the bacteria. And next type of food is that is called the shellfish and finfish. The microbial flora of the fresh these both fishes, because they are the aquatic, and these are very largely reflection of the microbial quantity of the water where they are harvested, and of particular significance where the water is sewage polluted or sea or that we have harvested from the seafood as a seafood. So main bacteria that can be cause the spoilage, and that can be present, that is of Vibrio cholerae and some other species of the Vibrio like Vibrio parahemolyticus, that is the mainly causes the gastroenteritis and epidermic infections in different persons. Okay, and these seafoods sample that include the fish, shellfish, and other that grow in the contaminated water can concentrate viruses and may be source of the hepatitis infection, and like raw oysters, clam to polluted waters, and that causes the numerous epidemics in the various parts of the world. Next, that is very much important, is called the milk. So at the time of we drawn the milk by the udder of the healthy animal, the milk contains organisms that have entered the teat canal through the teat opening. Okay, so they are mechanically flushed out by during the milking process, and the number present at the time of milking has been reported to the range between the several hundred to several thousand millimeter. The count vary among the cow to cow and the person who are handling the cow. That is also depends on that. Okay, and from the time of the milk leaf, the under the when we dispenses to the container, everything which come into contact with the potential source of the microorganism, and that includes the air contamination that is comes from the environment, and milking equipment that we are using for the milking process, and some other the handlers personal hygiene and sanitary condition, that also depends, and that also causes the milk spoils rapidly. Okay, so milking is generally performed under the hygienic condition, very strict attention to the sanitary practices, and that will result in the product of low bacterial content and good keeping qualities. Okay, next we can say about that basis of their major characteristics, that is called the biochemical types we can check, temperature resources we can check, and ability to cause the infection and the disease that we can do. Okay. Next we comes on the cereals, that bacteria that in the wheat flour, that include the bacillus, coliform bacteria, some other bacteria like Sarcina, Micrococcus, Flavobacterium, Serratia, that all causes the contamination in the cereals also. And sometimes the molds are also involved. The mold spores are chiefly of the Aspergillus and Penicillium, and with Cladosporium and some other genera, and Aspergillus flavus and Aspergillus aflatoxins, that produces the aflatoxins also. Okay, about that, different type of flours have a different type of contamination. The cornmeal flour that contains several hundred to several thousand bacteria and molds per gram, and Fusobacterium and Penicillium, that also causes. And because of the incubation of the moist condition like malt containing the high number of the bacteria, and the surface of freshly baked bread that is free from all viable microorganisms. But when we put it on the some room temperature, that can be contaminated by the mold spore, by the air during the cooling and before wrapping, and the cakes are similar subject to contamination, and spores are when with the appropriate conditions is there, so spores can be germinated to the vegetative cells and they will produce the spoilage of the food. And the contamination of the cereal grains, that is also affected by the different type of molds, because the molds contain the different type of mycotoxin that is responsible for the different type of disease in the human being. So about that, first we have discussed about what kind of organism, what kind of food, and what type of contamination they are producing. Secondly, we will discuss about the spoilage of the food. So, considering the variety of natural food substances and method by which we are using, the handling method which we are using, so different type of organism and different kind of microorganisms are potentially contaminant, and the type of food substance and the method by which it is processed and the preservation method can be used for the different organism to prevent from the spoilage. Okay. So most foodstuffs serve as a good media for the growth of many different microorganisms, and they will give the chance to grow the organism that will produce the that will produce and change the flavor, odor, and qualities of food. So they will degenerate the substance and they will produce some certain type of substances, like in the putrefaction process, the protein food, that proteolytic microorganism that will change into the ammonic acid, ammonia, and amines and some hydrogen sulfide, and some this sulfur molecules that is responsible for the smell and putrefaction process. Next process that is called the fermentation, that is caused by the microorganism in which the organism microorganism will effect on the carbohydrate food, and carbohydrate fermentation microorganisms that will change into the acid and alcohol and gases. Third type of process that is called the rancidity. The rancidity is a process in which the fatty food and lipolytic microorganisms work on it, and that will change into the fatty acids and glycerol. The changes that microbes that can cause the food that are not limited to the result of degradation. They may also be caused by the product of microbial synthesis. Some microorganisms that is discolored food as a result of pigment production. And slimes that may be developed in or on food by the microorganism that is capable to synthesize the certain polysaccharides also. So if we want to discuss about the fresh food, so how they spoil, what are the organisms, so we can discuss here in short. So first organism we can say that is a bread, and the type of spoilage is moldy, and main organism that is involved are the rhizopus and penicillin and agar, that is aspergillus. Okay, and maple syrup and syrup that is destroyed by the anthrobase bacteria, Saccharomyces and Zygomyces, and the most organism that is responsible is that is saprophytic molds and yeast, and type of spoilage is ropy and yeasty. They are pink, they change this into the pink and moldy, and all these things we can prevent by the different methods. About the cereals, we can use the low moisture content, so little difficult to preserve the growth of microbes, but we can prolong the deterioration and spoilage. And the mainly the temperature on which we can store the food is on the 4.4 to 7.2 degree. Okay, and some other product like bakery products like bread, rolls, cakes, and pastries that have enough moisture, so these foods are easily and they are good source. They are good providing the good energy source and food to the microbes. So they are the main effective foods for the microorganisms. So we have to give something to prevent this. Okay. Next type of food we can include is that pickle and fresh meat. About the pickle namely the type of spoilage is film or the yeast. Sometimes you are find out that some yeast kind of film is made on the pickles, and on the fresh meat putrefaction is there, and main organism that is responsible is a Aspergillus niger, Rhodotorula. And about the meat putrefaction, there is an Clostridium, Proteus vulgaris, and some species of the Pseudomonas. Next type of food is that is called the cod meat, that is of spoilage is about the molting, and main organism that cause the spoilage is Aspergillus, Rhizopus, and Penicillin, and generally they have producing organism has producing a greenish or the slime like substance that is caused by the lactobacilli and some micrococcus also. Okay. Next type of organism we can say that fish. Fish is of spoilage is like a discoloration, putrefaction, and the organism which is responsible for that is Pseudomonas and some Flavobacterium. Next food is called as axe, that is of green rod, spoilages and colorless rod, and the organism which is responsible is the Pseudomonas and some Proteus that causes a black rods. Okay. And concentrated organic juices, they will change into the off flavor. That is due to the presence and growth of lactobacilli and some acetobacter. And other food like we can say this is the poultry. Poultry that will change into the slime and odor. So that these organisms like Pseudomonas and some allifigens are responsible for the this type of putrefaction. Okay. So this is about the all fresh food and their contamination and their spoilage. Before studying about the prevention, we should have an idea about the canned food spoilage also. So the different type of product like low and medium acid products, and the products that pH level is about 4.6 like corn, peas, spinach, asparagus. So the type of spoilage that is of flat sore, okay, that is produced by the Bacillus species, Thermophilus, another Clostridium species, sulfide spoilage, and aerobic spoilage. So cane how you can identify the in the cane possible loss of the vacuum on the storage. Okay, and content of cane appears not usually altered, pH markedly lower, sour, may have a slightly abnormal odor, and sometimes cloudy liquors may be present. And second type of food is that is called the acid products like pH lowered from 4.6 like tomato, juicy fruits and fruit juices, and the type of spoilage is flat sore that is caused by the Bacillus thermophilus and Butyricus anaerobis, Clostridium butyricum, some yeast and some molds. And we can see in the cane is swell, maybe burst, can swell, usually burst, and can swell, maybe burst, and sometimes cane will be changed into the flat. Okay. Fermented butyric waters, and what will change? Acid odor, fermented yeasty odor, and surface growth is musty odor. So this type of changes that can be present in the different type of food. So the cane food, about the cane food, because of the heat resistant spore formers like Clostridium and Bacillus that constitute the most important group of the microorganism in the canning industry. And the three most important type of microorganism spoilage of the commercially canned food are flat sore spoilage, and thermophilic anaerobic, that is called the T spoilage, and third one is called as putrefaction. Three type of spoilage is there that is produced in the cane food. Okay, and this that all involves the spoilage of cane food together with a change in their products. Okay, so uh, when we examine for the microbiologically is all these cane foods, so we will obtain something. And so first we will discuss how we can prevent it. So the main thing is asepsis, that is very much important. That is sanitize the equipment, that is maybe source of many bacteria, and they mainly forming bacteria that causes a so sourceness of the bread, cake and other baked products, and that can be stored by the different type of molds, and that is contaminated by the mold spore. How they can prevent? By the use of heat. We can use the heat in the different variety to prevent the all kind of contamination, and for the all kind of we can say that spoilage. Okay, so mainly the food preservation generally which we are doing in the refrigerator, deep freezer, and sometimes we are using the temperature also. Nowadays we are also using some lyophilization technique. So the modern method of the food preservation, that is mainly based on the aseptic handling and high temperature, that is of boiling, and high temperature that also include the steam under the pressure, pasteurization, sterilization of the milk and aseptic process. And next is the low temperature by the refrigeration, freezing, and by the dehydration, by the osmotic pressure, by in the osmotic pressure the concentrated sugar and with a brine. And in chemical, we can also use the organic acid substances that developing during the process, that is called the smoking, and substances that is contributing by the microbial fermentation, that is acid. And next method we can also use for the preservation is the ultraviolet light and ionizing radiation for the preservation of different type of. So first is the use of different type of heat. So the high temperature is the one of the safest and most reliable method of the food preservation, because heat is a widely used to destroy the organism in the food products like in cans, jar and other type of conditions that restrict the entrance of microorganisms after the processing. So the steam under pressure, such as pressure cooker, is most effective method for the high temperature food preservation, and that will kill the all vegetative and the spore organisms. Okay, and the food preservation by the heat, that is required the knowledge about the what kind of food it is there, what kind of bacteria can be present. So that depends on the knowledge of all these things, and we have to have an idea about that this food is containing spores or not. Okay. In addition, one must consider the rate which the heat penetrates the food of the different consistencies, about their size of container and type of food. So the killing of microorganisms by the heat, that involves some time temperature relationship and products that we are using for the sterilization. Okay, and generally baking product may be the solid. So some products are in liquid form. So lot of things are there, but before processing we have to just have an idea about the all kind of things. Next type of method we can also use it as a canning method. The temperature used for the canning food range almost about the 100 degree centigrade to 125 degree centigrade, and for the low acidic food. And the canning process does not guarantee that style product. Okay, so sometimes spores may be present that can be germinated. Mostly the important organism is the Clostridium botulinum. About the milk, so most important thing that is called the pasteurization. So the pasteurization that is fixed quantity and a fixed temperature of on which we can put the substance, and after that suddenly or immediate cooling we will follow, so by which due to the osmotic differentiation of the organism, the organism will kill. Okay. So for this we are using the holding method and flash method. Okay. So by the pasteurization technique we can sterilize the milk products and beer something else, some other product also. Okay. So by which this method some resistant kind of organisms cannot destroy, like Mycobacterium tuberculosis and some species of the Clostridia also. Okay. And aseptic processing, that is relatively new commercial development in the food industry, that is most acceptable. Then in this the food item is commercially sterilized and packed into the previously sterilized container under the aseptic condition. Okay. In this process, they have this advantage that we are the container which we are using, that is good other than clean, and that is that provides a significant economics and use of different things. Okay. Sometime for the prevention of the spoilage we are using low temperature also, and sometime we will use dehydration procedure in which we will remove their moisture, so the microorganisms are unable to grow in that medium. Sometimes we will destroy their osmotic pressure, and after disturbance of the osmotic pressure, organism will cannot retain or cannot able to survive in that medium, so organism will die. In addition to the for the preservation we are using sometimes some chemicals for preservation, and like pickle we are using some substances, in some sauces we are using sugar and this also salt also, and sometimes different radiations are also used, like ultraviolet radiations are mainly important. Okay. So these are the methods by which we can prevent this. You can see that is the use of low temperature. So use of different type of chemicals in the bread and different type of things. We can use the different type of radiation like ultraviolet light, that is also in the bakery, that is a good one. Okay. As we discussed previously, the spoilage is also very important. So we have to have an idea about what kind of things are there, what kind of spoilage that we have discussed in previously in same lecture. And after learning about the spoilage, we have just given you an idea about that how we can prevent all these things. Okay. So in the today's lecture, we have just given an idea. I tried to give you some brief introduction about what kind of contamination, what kind of preservation, and what kind of spoilage the food is generally facing, and what kind of organisms is responsible for all these things. So I hope you are able to understand all the facts. So thank you very much for being with me.